afternoon we start with the project, and we need to know exactly if the list of people that we have is the same as the list of people, as the people that are here. So we are going to circulate after the first coffee break a list with all the names we have. If you see your name, just cross yourself. And if you don't, write it down, okay? So then we can see if someone that was registered didn't come, or if someone could only be here the first week, and then we can have the list of people to make the groups, okay? That's gonna be in the coffee break. I need to print it out, basically. And I think that's the only announcement I had to make. Yes, that's the only announcement I had to make. Okay, let's start then. I think it's time. I don't want to be late. Okay, so as I told you in the email I sent last night, we did a small change in the schedule. So I'm going to teach first, and then Veronica is going to teach second, and then in the afternoon we'll have the projects. And I thought I would be showing you a little bit of how you can think about animal movement as a pattern forming process, more or less, which I'm going to defend now in a while, why I think so. And especially what are the implications that it has for population dynamics that, as we saw last week, a lot of people think about that as a pattern formation process, right? And the reason why I think movement is an important component of population dynamics, well, I think no, it is an important component of population dynamics. And the reason why I wanted you to think about that is because, as it will become clear in a while, it's one of the key assumptions in a lot of ecological theory that you had been seeing last week and that you probably will see around in the coming steps of your career. So the work, what I'm going to do, I didn't want to present a seminar, although I only have one lecture because I thought a seminar would be boring. And I'm going to do a combination between showing you the things that we do and putting them in context with proper theoretical background. I'm going to sketch two very simple calculations. I'm going to give you some hand-wavy arguments to defend some of my statements. And if you want more formalism, we can chat about that in the coffee breaks, and I can also extend a bit more into anything that you may come up with. So besides this work, we also do a lot of work on microbial pattern formation. So we work with bacterial biofilms and with slide modes. If you're interested in that, we can also talk at any point. And we also do a lot of work on vegetation pattern formation, both from the perspective that Johan was introducing last week, and also looking at how these patterns form from interactions between pairs of plants, and trying to do some models for how the root system develops and how it's rearranged when there are neighbors around. I'm not going to show you anything of that because it's been a while since we don't do anything on microbes, and because I think Johan put the level pretty high in terms of vegetation patterns last week, so it would be a bit stupid from my side to compete with that. So regarding the new work, the hero of this story is this guy that you see in the screen. The other one with the white t-shirt is my PhD student, Rafael. No, I'm kidding. Most of the things I'm going to show you are Rafael's PhD thesis, who is about to finish. And I'm not going to show you anything specific on Benjamin's work, who is here in the audience, but his work was really helpful trying to formalize these ideas and trying to get an understanding of why we were getting things right in simulations. No? So we were getting computationally things the way we thought they were, but we didn't know very well why. Actually, I'm going to tell you a very funny story at the end on how a paper that is not fully correct can be very interesting and open a lot of years of research, just getting it right. So I thought, yeah, credit to him was also due. So let me start showing you a data set that is very similar to some data sets you've seen already. So what you see now in the screen is the growth curve of an E. coli culture over time. It's very simple. In the y-axis, you have the concentration of cells, and in the x-axis, you have time. You see that, not surprising for you at this point, I hope, the population first grows and then saturates. To me, this is a pattern already. I mean, we've been discussing a lot about Turing patterns and spiral waves, spatiotemporal chaos, excitable systems, and a lot of cool stuff. But that, to me, is a pattern. And it's the simplest pattern I can come up with. Still, I'm going to show you that understanding this pattern, or better said, understanding this process and the variety of patterns that can come up with here is an extremely challenging problem, both computationally and empirically. My lecture is going to be a conversation. I'm going to show you data, then I'm going to try to do some calculations. I'm going to introduce a lot of typos, then you at home can correct them, and I'm going to go back and forth between theory and data. So these are the conditions in which these experiments are usually done. The bacteria are in some growth media. They grow, and then you can measure the concentration of cells. Last week, we learned how to do a model for this system that accounts for the individual level processes that are taking place there, right? Nigel taught us how to do that. So we can think that we have our cells in some kind of growth media, and they are undergoing a series of reactions. The minimum set of reactions that we can have in this system is the 3 that you see in the screen. So basically, cells can divide at a given rate. They die at a given rate as well. And then we had some discussion last week on which is the way of introducing this limitation of growth. And at the end, we decided, or Nigel told us, that this kind of weak constraint for population size was convenient theoretically. So far, there is nothing new there. If I start with this set of reactions, I can write a master equation for the number of cells that I have in my system that I'm going to write here because it's going to be important in a while. So I can write I'm going to skip all the steps, and I'm going to go all the way to the master equation just for the sake of time, and because we already learned how to do this last week. But I am going to write the rates explicitly. So I'm not going to work with transition rates or with step operators. I'm going to write for you the specific form that each of the rates have. Well, let me put here another n, and that's going to be easier. So this is the master equation for these individual level reactions. You see that this is a balance equation for the probability of having n cells at a time t. And we have some terms that are the inflow and some terms that are the outflow. This is just a balance equation as we discussed last week. So in principle, with all the tools that we learned last week, from here, yes? Thank you. So from here, we already know how to go to a macroscopic theory, right? And we learn how to do the Van Kampen's expansion of this master equation. We know that if we consider different terms in that expansion, we get only the deterministic part of the equation, we can get the fluctuations, and we can learn many corrections to the deterministic dynamics. The simplest thing that we can do is to stay with the deterministic limit, and then we have the logistic equation that probably you are all bored with it. I mean, it's probably the equation that you've seen more times. I don't know if in your life, but at least in the last days. This is nice. It gives me some information. It's easy to fit without going very fancy. I'm not an expert on statistics, so it's just easy to fit the parameters using whatever method you want. And it gives us the best fitting model, and we can get some information into what is the maximum population size that I should expect at equilibrium. Of course, this is all deterministic. If I am accounting for fluctuations, I will have fluctuations around my current capacity, and I will have a distribution at every time step. Right? I'm just keeping things deterministic so far. Everyone is okay up to here? Just reactivating neurons after your cultural route over the weekend? Yes? Okay, perfect. Museums are very demanding. Okay, so who is happy with this thing I've done? Oh, thank you, Facundo. If not, okay, good, good, good. Who is unhappy? No one is unhappy? Okay, that's a problem. Anyway, this thing I've done is good, it's reasonable, and it's very reasonable for the system that I had, right? Because I had my bacteria that were in a growth media, they were all moving around, and it makes sense that my description of the system is just the number of bacteria that I have in the population or the concentration if I go to a density field. But if I think a little carefully about what I've done here, I've done a very, very strong assumption. Who can tell me what is that strong assumption? I'm going to ask a lot. I mean, if you were bothered by how much people were asking last week, today is going to be crazy. Yes, Ian. I assume well mixed. Where did I assume well mixed? I did. It was on purpose that I throw the master equation for you because we are all very comfortable with this kind of equation. We've written it so many times that sometimes we do it in automatic. Fine. But what I did here, if I think carefully about what I did, it's actually quite interesting, right? I took this competition term, and basically, the way I could think about writing this master equation, I could say, well, the probability that I have end particles at time t plus dt is equal to the probability that I have to me, bacteria are particles, so maybe I change things a little bit. So anytime I take a particle, it's animal, bacteria, it's whatever. So it's the probability that I have n at time t, and nothing happens. Let me write it properly with all the terms. So 1 minus the probability that one dies, minus the probability that one dies, just by natural death, plus the probability that one dies due to competition. And then I have the probability that I am in a state with one more particle, and one particle dies, plus the probability that I am in a state with one less particle, and one reproduces, plus, again, I have the probability that I am in n plus 1 state at time t, and one dies because of competition. And I'm not writing here anything by now. By the way, when I say that the probability that one reproduces is equal to this, is because I'm assuming that dt is very small. Okay? And I'm doing an expansion. I'm assuming that the probability of having a division, the probability per unit time is constant, so the rate is constant, and I would need to do the proper, this is the probability of having, of not having reproductions between t and dt. I would need to write that probability explicitly, and then if dt is small, I do the expansion. Think about that and try to do it as homework, and if you don't come up with how do I get here, we can discuss, but I want you to think about that. Anyway, when I write in these probabilities, the ones that I didn't write on purpose, what I'm saying is that this is a nonlinear process, so basically I'm describing it as the collision between two bacteria. And I'm saying that the probability that that will happen is gamma divided by omega, which is my rate. And then we can think about this process like this. So imagine that I have only 4 bacteria. And I'm saying I'm going to first pick my focal bacteria to be this one. And she will be able to interact with this, or with this. If I repeat that for any bacteria, is when I get this term, and then if I assume that they are, I don't want to count pairs twice, is when I divide by 2. So this is what I am assuming when I write this term here. Basically, I'm assuming that any bacteria in my population can collide with any other in the population. And collision is a pictorial way of introducing, for example, competition for a limiting resource. Let's say there is only one there's only one final piece of cake, and if 2 of the bacteria go for that piece of cake, only one gets it and the other one dies, more or less. That's the picture I'm following. This assumption is reasonable in which conditions? But what does it mean? What does it mean well mixed? So if I think about, I introduced already a time scale here, right? Is dt, is this time at which I will only have one event. I have my continuous time and discretizing it in small times dt. This dt has to be much smaller than all the times that are defined by the demographic rates, right? In principle, it should be much smaller than b times n plus d times n plus omega plus gamma over omega n times n minus 1 and the inverse of all that huge rate, right? Let me say that this in principle has to be much smaller than this time. The other ones we'll assume this is the leading term in the rates, okay? This is necessary for me to make sure that I only have one bacteria dividing, I only have one event in this dt. But when I am writing this here, I'm also assuming something else. I'm assuming that this dt has to be much larger than what? Than one time. Let me use tau, so then I can follow my notes when I get lost. What is this tau? Collision time? Which type of collision? The time they get together. We have here one idea. Any other idea? So if I am counting all these pairs, this time has to be the time at which all these events are possible, right? It has to be the time at which my population mixes. So if you think that these particles or these bacteria are doing diffusion, this time should be approximately equal to what? Which is defining the mixing time scale if I have a bunch of particles moving, one particle doing Brownian motion around? The diffusion coefficient and the scale. Everyone agrees with that? Size squared divided by d, more or less. This is a hand-wavy argument, but it's just to set a little bit the idea that if I want this logistic model to work, and if I want this master equation to be a real representation of my system, L squared divided by D has to be much smaller than, right? If not, then things are start to get a little bit crazy. So there is one case in which this may not be true, which is when, for example, if my diffusion is very small compared to the interaction rates, then this assumption doesn't hold. But there is one other case in which this doesn't work, at least one that I can come up with. I'm going to show you a movie, and then you tell me, what do you think? So this movie that many of you are so tired of seeing it, I promise you, yesterday I tried to update it, but I couldn't because I didn't find any other. It's a pity. What you're going to see is a population of wolves in Yellowstone that have been collared. That means that they have a GPS. So more or less we did with this, or these people from the wolf project did with this wolf the same thing that Samsung does with us, or Ma, or Apple, or whatever. They put a GPS on us. And this is how the trajectories look like. Well, you can see, no, the colors even with the light on. And I could be tracking these animals forever, and ever, and ever, and ever. And the spatial structure of this population would be this. So there is no way that I define any time scale here that my population is well mixed. It's just not defined. There is no, I cannot define a mixing time globally in my system. And there is no limit in which I can define that quadratic term or law of mass action for my reactions. It's just impossible because of the way my animals are moving. They don't like each other, basically. So then they will never mix. So this is already an interesting. It's an interesting observation that opens us the door to do a lot of modeling work, trying to account for how we can rewrite basically those master equations when we have this spatial structure. And that's something we've been trying to do for a lot of time, and well, I'm going to show you where we are. It's been a lot of fun, but also a lot of frustration from time to time. In any case, this opens the door for us to having to quantify animal movement properly. Ah, you had a question? Yes. Yeah, that's a good question. So this system is way more complex than what it looks like here. So wolves, they like to live in groups, in packs, in families. And they are super happy when they are together with their friends, with their family, with their pack. But they don't like other packs. So what you are seeing here is one trajectory from trajectories from wolves from different packs. So white is one animal, yellow is one animal, so on and so forth. But that doesn't mean that the white is not surrounded by anyone. The white will be surrounded with other wolves in its pack. It's just that we are not tracking them. So if we were tracking the whole population, that's a very interesting problem of working with these systems that is really hard to track all the animals. Then you would see that the white would be a cloud of trajectories here, a cloud of trajectories here, a cloud here, a cloud here. There will be a hierarchical structure in the population. Anyway, so this complexity that we saw in movement already tells us that quantifying the ways and the way animals move is important. And that's the way people usually does this kind of thing, which is coloring animals. And then for some time, we have their trajectories. It's a very interesting data set to try to understand how animals interact with the environment, how they decide to allocate into different tasks during the day, and so on and so forth. So this is an example of a gazelle living in Mongolia. This is the system I used in my PhD. And this is how one of these trajectories look like. You already see a lot of interesting things. The first one is that the data is very ugly, in the sense that, well, it's not very clear here, but I can tell you. The sampling rate is not constant. Yes?
If you track the alpha of alpha male of the pack, or a scouter wolf, or some other type of member of the wolf society, there will be a different trajectory?
Well, every animal will have a different trajectory because there is individual variation.
I mean the statistical pattern that emerges, there I guess is a Levy walk, for example.
So to be honest, in that case, I don't know if any of those animals was the alpha male, but you do see differences in the trajectories of males and females, juveniles and adults. You also see differences depending on breeding season and non-breeding season, and that's the power of this data set, that you can use the data set, for example, to try to infer behavioral modes, for example. Yes?
I guess with wolves, at least packs move together, so I think treating them as individuals is a very good approximation.
That's one reason for only tracking one animal within the pack. If you are interested in social interactions within the pack, there is some people that study animal societies, and they pack They track single entire packs to try to understand, for example, hierarchical relationships. But yes, as a zero order step, it's a pretty good idea. Anyway, so here you don't see that the sampling rate is very uneven and there are missing points because of the representation. But for example, if you plot the x coordinate versus time, you will see that I don't have a point, let's say, every day or something like that. It's like a completely heterogeneous sampling rate. And that's done on purpose because you want to try to understand more or less how animals move at different time scales. And that's what I'm going to show you in a little bit. So the problem of this visualization is that it is not very informative because you see the trajectory there and it's really hard to get information from it, no? But what you can do is you can think that this movement, these trajectories, are just realizations of some stochastic dynamics that is governing the way animals move. So this is the realization, the single realization of a stochastic process, let's say, and it has some underlying statistical properties, as was already asked around there. So what is really interesting is to try to obtain the statistical properties of this system. And I can tell you something that is very useful to perform this analysis, which is that when you look at the distribution of the velocities of the animal, it follows a normal distribution. So why do you think that's important, or helpful, or useful? Well, we like Gaussian processes in general, right? Most importantly, we like them because we can describe them using very few moments from the distribution, right? So from a Gaussian process, if I know the mean, if I know the first and the second moment, or the first and the second cumulant, depending on how you like to think about distributions, I have all the information about the process. So in principle, if for this process I know the mean and I know the autocorrelation function over time of the position of the animal, I know how to reconstruct my stochastic process as knowing that it's Gaussian. And that's something that is always super important to test, that the process is Gaussian. I know some people talk about animal movement in terms of Levy flights and power law in step lengths. I have very strong feelings and opinions about those that I'm not going to say here because I'm being recorded. But with 1 or 2 beers and half of caipirinhas, I'm super happy to discuss those. But remember, the ratio between beers and caipirinhas is always 1:2, okay? Just to keep mental brightness. So how would you try to measure the structure of this trajectory? What is the thing that you would compute here? I'm going to tell you already something. The mean is not important. My animal has some region that she likes, and she's just doing some excursions around that mean. There are some animals that don't do that. For example, the one that migrate. They are going from one place to the other. We are going to focus on animals that like to live around one area, and they spend all their time there. Well, I just told you, no? I want to compute the autocorrelation function. But instead of computing the autocorrelation function, I'm going to compute the semivariance function, which is the same. I'm going to write the definitions, but it has best statistical estimators. That's the only reason why I'm going to choose it, okay? Because the statistical estimators for the semivariance function are better than for the autocorrelation function. Yes, question? The GPS is sending a coordinate every once in a while. I don't have like a smooth curve. Every once in a while, the GPS sends, the device that the animal has sends a coordinate to the satellite, and that's all I record. And that's what I meant by uneven sampling rate. The frequency, the time between emissions is not constant, okay? And sometimes the device stops working and then comes back after 4 days, and so it's a messy data set, okay? It's not an easy data set to work with, which makes it fun. Anyway, let me write for you the definitions of the autocorrelation and the semivariance that I'm going to use, just to avoid any inconsistency between the slides and the notes. So in principle, if my process is not stationary, then the autocorrelation depends on the two times, not on the difference between them. And I already changed things. This is the semivariance. And this is going to be x t minus x t prime square. You can define the same for in gamma, and then you can do the radial average. T prime, that would be equal to 1/2 plus gamma y. So this is kind of this would be the radial semivariance function that is, I'm going to define it as an average between the x and the y coordinate, and basically tells me between two times, t and t prime, what is the radial distance that this animal disperses. Okay? I'm going to open some of the calculations in a while. All the calculations I'm going to open are going to be in 1D, just to make things easier and faster. But doing it in 2D is the same. You only have to repeat it, changing x by y, and then whenever there is a radial, you do this, okay? In case you want to open calculations at home. And then the autocorrelation function that I'm going to define with sigma, t prime. A good exercise for you to learn all these definitions. I have a horrible memory, and I always had a bad time memorizing all these definitions. So a good exercise for you is to find, unless you are already familiar with it, in which case you can repeat it, because you will probably don't get it right the first time, is to get the relationship between these two things. It's super easy. You just expand the terms and then rewrite, and you will see what is the relationship. But it's good, because I'm going to change one and the other, between one and the other, and it's good that you have your brain set up for these transformations. So one thing that is interesting and important to know about these two ways of measuring correlations within the trajectories is that the autocorrelation, as my times, at least for the processes that I'm going to introduce in a while, as the times are farther apart from each other, the autocorrelation is going to be a decaying function of the distance between times, whereas the variance, the semivariance function is going to be an increasing function of time. Right? Just because one is telling me directly how much 2 points in time are related to each other, and the other one is telling me how much distance the animal is dispersing, right? So it makes sense intuitively. So let's practice a little bit these definitions. Imagine that I have Brownian motion in 1D. That I can define by this Langevin equation with these 2 times correlation function, and of course the mean is going to be 0. I already told you that I don't care about means, so the mean is going to be 0. If I want to compute this function for the stochastic process, I think many of you have done this probably already, but basically, the first thing that you have to do is I have to integrate this equation and then you can apply the formula, the definition, sorry, of the semivariance. And after doing some homework, and basically you will get here the square of an integral that you have to write as a double integral, and so on and so forth, you will get that the semivariance function of the Brownian motion increases linearly with the time difference. This is probably a result that if you've done statistical mechanics, you are bored of seeing, so I'm not going to repeat it. But this is what you get if I get Brownian motion. What is the meaning of this result? The meaning of this result is that if I have an animal that performs Brownian motion and I throw it at a given coordinate, she will start moving around, and as time goes by, the mean square displacement of that particle is going to grow linearly with time. In principle, I would say that for this trajectory, this looks like a good first hypothesis, right? Well, let's see what happens if they follow Brownian motion. I'm cheating a little bit because I already showed you the Wolf dataset, and you know that that's not possible. But let's see what is the result that I get if I work with this null hypothesis. So what you're going to see now in the screen in a while is this semivariance function that I computed for Brownian motion, and it's going to be plot on top of the semivariance function that I computed for the dataset of this trajectory. And this is what you see. So the blue dots is the dataset. So this is how the empirical semivariance function of the animal looks like, that usually you will see it in the literature as semivariogram. This is the semivariance as a function of the lag. The lag is the distance between two time points. And the red line is the Brownian motion. You see that the fit is horrible. And it's horrible for something that we already kind of knew from the wolves, even though this is a gazelle and they are different animals. I hope we all agree on that. But the main key difference that we already see is that the semivariance of this gazelle, it saturates, it plateaus. It doesn't grow indefinitely. And this saturation is what is going to make well-mixing models very, very bad, at least for terrestrial mammals, which are the ones we are interested in. So how would you correct this? How would you propose a model? What do you think I'm missing with my models that will help me correct this thing? Space dependence. That would be a good idea, space dependent of the diffusion. If I were a physicist, I would assume that my animals have some potential that is not allowing them to move around freely. They are confined in some potential. They have some attraction towards the center. That's why they are not all over the place, which makes sense. I have an attraction towards Spain. And if I am a physicist, I would assume that that potential is a harmonic potential, just because if I'm a physicist, then I am allowed to do that, right? And that is a way of refining that model. That model is now the Austen-Uhlenbeck. How many of you are familiar with the Austen-Uhlenbeck process, stochastic process? Okay. So then if you are familiar with the Austen-Uhlenbeck process, if you're familiar with the Austen-Uhlenbeck process, this is not new for you. For those of you that are not familiar, the Austen-Uhlenbeck process basically has two different components for the evolution of the position of the animal, in this case, or for the variable. One is a deterministic drift, a linear attraction force, that is dragging my animal towards its center of life, towards its nest, if you want. And then, it has, again, a random force that usually in this context, we think about it as some other component of the movement that we cannot resolve. For example, this is an attraction to go back to the home brain center, and this could be excursions that the animal does to take food, to look for mates, or anything else that you can think of. This the two-time correlation function of this random force is defined like this, and this is just to make sure that the Austen-Uhlenbeck process, when I take this time, the inverse of this time going to 0 plus this sigma, which is a variance going to infinity, if I take the limit of this going to 0 and this going to infinity with the ratio being constant, I recover the Brownian motion. Okay, so you can think that if this particle is very, very strongly attracted towards the center, but at the same time has a very, very high dispersal, in a way that both of them, the ratio between both of them is constant, then I recover Brownian motion. That's why this is introduced like this. With this definition, you can calculate everything. You can calculate the two-time correlation function of x, not of t. And you will see that now. Let me just, for clarity, use the lag here already. So the two times correlation function of the Austen-Uhlenbeck process decays exponentially. This is the first important result. The position of the animals now are correlated, and they are correlated because of this drift form here. And you can also compute the variance, the semi-variance function. That has this form. You can work with this, you can do the math at home or in the hotel, and if you have any problem, you can ask me if you want to. I'm speaking a bit slowly, so if I open the calculations, I won't have time to show you all I want to show. Anyway, what is important about this is that, first of all, the correlation decays exponentially, and because of that, the variance of my trajectories also saturates with time. Right, which is already something that I wanted we observe in the data. No? So we can now fit this model to our data set, and it's much better. How many of you think this fit is great, is perfect, or very good? Would you go further into this, or would you be happy with this? More or less here. If you are happy with this, you will enjoy the rest of the class or the lecture. If you are not happy, then you will have a lot of questions at the end, which I hope you also have. It's a very decent data. It's a very decent fit, sorry. We are capturing the main thing, which was that our animals are confined. And what is more interesting is that we can give a meaning to this saturation spatial scale. I'm going to go into that in a little bit. But first, I want to leave a little bit the route of the rest of the lecture to tell you that this fitting is very good, but we can improve it. Yes?
But this data is only for a single individual, right?
Yes.
So can you really trust the statistics that these
Okay, yeah, that's
give?
a great point. That's a great question. There are two things that you can do here. So first of all is that sometimes, not sometimes, for this specific field, you are interested in the individual behavior. So what you need, your way of getting a statistical accuracy, if you want, or statistical strength, is by tracking the animals longer and with higher resolution times. If you want to ask questions about what is this scale for gazelles overall, then you need to track a lot of animals. You need to do this, and you need to do a statistics over these parameter values that you get. But it is very, very often that you want to understand specific behavior of the animals, no? In any case, you always start with one trajectory, you perform this for one trajectory, and then you do statistics over the fittings that you have for trajectories. But that's a great point. Something that this field misses a lot is tracking a lot of animals at the same time, because putting these colors is expensive and it's very time demanding. So usually you have very, very little population coverage, which is something that is very problematic for some of the things I'm going to say in a bit.
So, and if you add a repulsion between, like the wolves, it will have the same effect of this saturation?
Yeah, you have you go fast. So if all the models I'm going to show you, if you consider a lot of animals, they will assume that animals are independent. They are moving independent to each other. I'm not going to introduce here. So if this were x1, and I write a Langevin equation for x2, the covariance matrix of the x1, x2 process will always be diagonal. I will never have x1, x2 correlations. But you can do that, yeah, you can do that. And sometimes all the statistical estimators that we have assume independency. And sometimes they are horrible. They are just crap because trajectories are not independent, especially for the things that we want to look at, yeah. Yes?
I would think that the stage of life of one of these animals is really important for this curve.
Mm-hmm.
This is characteristic for adult or no?
So this specific data set, I don't know if it's an adult or a juvenile. My guess is that it will be an adult for sure. And I don't know if a male or a female. But yeah, sometimes you have males have larger area requirements than females. It also depends on the quality of the landscape. If you throw these gazelles, if you track them in a year where they have a lot of grass, maybe this area is smaller. But if they don't have so much grass, it's larger because they have to look for larger areas. Predators have larger areas than prey, and a lot of there is a lot of ecological theory hidden behind these trajectories. But yeah, you're right. It depends. If I track a female when she's in the breeding season, and when she's not in the breeding season, or if she has calves around, it's completely different.
And the other question I have is, this is around a year, more or less, of lag.
Yes,
and
a
this
little bit
is
less.
Small compared to the life of the animal, no?
Yes.
Okay.
But something that is really important when you decide that how long are you going to track your animal is that in principle, if this already saturates, and you know the ecology of your animal, you have a pretty decent sampling time. If you sample short times, imagine, and when I plot my semivariance function, it stops here, then Brownian motion maybe is a good fitting, but that's just because I'm missing data, and that's something I want to show in a little bit. Yes?
Where do all of these error bars come from?
Where what? The error bars?
Yeah.
So usually, so this is the semivariance over lags, right? So you could, you can think that when I have smaller lags, I have more statistics, right? So imagine I have a year and I look at 2 hours, I have more data, and on the other extreme I have less data. So that's why the bars are larger, less statistics. It's the same as when you compute a radial correlation function, for example, in a material. Okay, so going back to David's question and to the things I was telling you, this fitting is good. It's pretty decent, but it's not as good as I would like it to be. You can see that here, the fitting is not that good, right? So basically, what I want to show you in a little bit is that only with one spatial and one time scale, I'm not able to capture all the complexities of animal movement. And to capture those complexities, and this is the challenging homework in case you want to get practice computing moments of Gaussian processes, which, by the way, I think is a very interesting task. It's a very good skill to have. This is a model that was introduced in this paper in 2014, and also tells us a very interesting story about conversation between physicists and ecologists. I didn't say that at the beginning, but I am a physicist by training, but I've been thinking with ecologists for more or less, I don't know, 10, 11 years now. So it's very interesting to see how they come up to the same place with different names, and this is a very good example of that. So if I'm telling you that I'm not able to recover the short lag behavior of this semi-variance function, what is that I might be missing, assuming Austen-Uhlenbeck process? If you do active matter, you have to know this. Bien, good. Sorry, that was my Spanish enthusiasm. So I told you that the position was correlated, right, with this model, but my velocities are not. I'm assuming that the velocities are independent. So if at short time scales I have a weird behavior, not weird, I have a different behavior from the one predicted by my model that I cannot capture, it might be because assuming that self-repulsion is not important here for a gazelle may be too much of an assumption. So to fix that issue, and this is the bonus homework track, the model that people usually use is called the Austen-Uhlenbeck with foraging. I'm going to tell you now in a while why it's foraging. And it has this autocorrelation function. And if you want to compute the variance, I'm going to give you a little bit of a spoiler here from your homework. You can compute it like this. So the way that you get to obtaining these autocorrelations is if you define now your movement model as coupled stochastic differential equations. So you would first have the Austen-Uhlenbeck, but now the stochastic force that is dragging, that is perturbing the linear drift is not white noise itself, but it is another Austen-Uhlenbeck process itself with some variance, 2 times correlation function given like this. So if you obtain these autocorrelation functions starting from these 2 equations, I can send you notes with this calculation open if you are interested, because it's not an easy calculation if you've never done this. You will obtain the autocorrelation function for this process, and just equating sigma u to the constants that you need, you will get what you need to have. What is important here is that now the velocity has another characteristic time scale. This tau f and tau h are 2 different time scales. One is defining the self-repulsion of the animal, and the other one is defining the relaxation to, let's say, its home range. Physically, what this means is very simple. I'm simply assuming that this force, that before was some sort of white noise that my animal was moving around, now it has self-repulsion. It's the only thing I'm accounting for. How important this is, it depends on the questions that you have. And this is always very important. If you're interested in long-time processes, maybe you can neglect this. But if not, maybe you cannot. But something that you need to be very careful is that when you feed, for example, this OU model to the data set, the spatial scale that you will get is different to the one you would get if you feed the proper model. So always be careful with these kind of things. Anyway, if I basically feed this gamma function to my data set, this is what I get. I get a much better agreement. And what is more important is that I get this agreement at 2 very different time scales. First, if I look at very short times, at very short lags, you will see that the OU process, the green light, is not able to capture this short time behavior of the data set, whereas the OUF it's able to do that. And at long time scales, if you look at the whole spectrum, as I already showed you before, you have a much better fitting. So what is the ecology that I can learn from all these calculations that I am doing? I can learn many things. The first one is that when I fit this model to my data set, I obtain ecologically interpretable parameters of the movement of the animal. So first of all, I obtain that this gazelle has a foraging time scale, if you want, or the correlation of the velocities has a characteristic time scale of about 6 hours. So for 6 hours, this gazelle is more or less moving in a ballistic way, if you want. It's moving straight with persistence. On the average of 10 weeks, more or less, she's crossing the total area at which she will live. So you can think that in 10 hours, she's relaxing more or less to this long-term movement behavior. And finally, I can also interpret the spatial scale, which is the saturation of this semivariance function, which if I assume that the distribution of the animal is a 2D Gaussian, and I define the area that this gazelle occupies as the 95th percentile of this distribution. So basically imagine that the animal has this distribution, and I assume this is a 2D Gaussian, and it's isotropic, and I assume many things, just to give you an estimate. If I recover the distribution and I limit the 95th percentile, the fact that the variance of the position is 4,500 square kilometers means that the area that this gazelle covers in a year is around 90,000 square kilometers. Just to give you an idea, that's more or less the size of Portugal. So just putting a color in this gazelle and fitting these stochastic processes to it, we get an idea of the relevant time and the spatial scales of the movement of my animal. I get an interpretation, a quantitative understanding of the segregated in space pattern that I was showing you before. I think that's very cool. It's very cool that just from a trajectory data set, a cloud of points in space, I'm able to tell you, look, these gazelles, at short scales, they have a 6 hours time scale, they cover the area of Portugal. This is really important ecologically. Yeah? Imagine that these animals live in the steppes of Mongolia, and imagine that you start building roads in a way that you don't leave 90,000 square kilometers free for them to move around. They will start having troubles.
9 or 90?
90,000. 90,000, yeah, it's the size of Portugal. If there is any error, it's here. Here is correct. If there is any typo, it's in the formula. In the result, no, because I check it. So this is really important in terms of managing populations, managing ecosystems. Here in Brazil, there is a lot of discussion about that, for example, with El Pantanal, with a lot of roads crossing natural habitats of different species, and so on and so forth. But let's go back to, most of you are interested in physics more than in ecology, so I'm not going to bore you with pretty pictures of animals. I'm going to do some calculations. Let's go back to our picture. Our initial question was, how is my description of the dynamics of a population changed when I need to correct this assumption, right? That was my point. All I've done with this detour that I thought was going to be 20 minutes and it's been 45 was to show you that this question is supported by empirical data. It's supported by what we see out there in animal populations. And this is really important. I mean, you will see these well-mixed models applied even to plants sometimes and to trees, which is, I think, a little bit, it's useful but complicated. Anyway, so now the question that we came up with is whether this complexity of animal movement is reflected at larger scales of organization. So it's reflected at the level of populations, the communities, the ecosystems, and so on and so forth. We've been working a little bit on that for the last few years, and I think all I'm going to show you now is slides. So let's build some simple cartoon about what does it mean being well-mixed and what does it mean being spatially segregated or having a finite spatial range that you occupy over space, over your life. So the way you could achieve this well-mixed scenario, in which here blue and gray mean probability of finding an animal, let's say, you can assume that your animal, as we were doing in the beginning, follows Brownian motion. And if, of course, if I don't have any boundary condition, then it will spread all over the place, so I need to confine it. And I will confine it with a smooth surface to make sure that the distribution is uniform. And on the other case, I have my two Oosten-Uhlenbeck processes. All I'm going to do now is with Oosten-Uhlenbeck, because when I have these self-proportion, things get way more complicated. So just OU. I'm just interested in the saturation in space. Then you have this segregation in space, and what is important is that if you look at the number of spatial scales that you have here to determine the interaction, it's very different. So when everything is well mixed, the only spatial scale that is set in the interaction is the size of your container. If you want, the size of your population. But as soon as you allow animals to have their own home range, then you have a lot of spatial scales. This is just geometrically, it's a way more complicated problem, because you have one spatial scale that sets the home range of the blue animal. Another spatial scale sets the home range of the gray animal. And another spatial scale sets the difference between them. Right? So now the interaction process is a complex process of all these three spatial scales. So this is probably the mathematically less rigorous slide I'm going to show you today, and the less rigorous that you will see probably in the school. But it's helpful to build some intuition, and I'm going to explain you why it is not rigorous. So the way I'm going to think about my animals, now that this separation of time scales doesn't work, is that I'm going to think that this gray guy is a predator, let's say, and this blue guy is a poor prey. And they are moving around, and my predator, that can be, for example, a lion, has some detection radius d that defines the area over which he will be able to kill the prey. So when they are this far apart, they don't interact, because the lion doesn't see the zebra and everything is fine. Well, for the zebra. When they are this close, then there is a probability that the lion will perceive the zebra and will eat her. How do I formalize this mathematically? I don't know very well, but how do we do that? I define the probability of having an encounter between time interval t and dt as the probability that I have a successful attack conditional to the distance being close enough, right? Everything so far, so good. If I assume that the probability of having a successful attack is independent from the probability of being close to each other, I can split this way. And now the probability that this distance is smaller than d, I can write it like this. So I can define the probability of having a successful attack as a constant, gamma. Let's say the lion is just it's easy, constant. I am not accounting for any small details. Gamma dt would be the probability of having a successful attack, and the probability that my distance is below the threshold, I'm writing it as the cumulative distribution function of the pairwise distance. And then this is what I'm going to call the encounter rate. This would be kind of the equivalent to the gamma divided by omega that I was writing before, but when I have a space. Why I was saying that this is not fully correct, if anyone is willing to tell me something? I'm doing a very brutal thing in this step here. That works in one limit, that we know, thanks to this paper here by Benjamin. So I am assuming that, so there is something very important here. These are trajectories. They are moving around. I'm looking at statistics in terms of the trajectories, and all of a sudden I'm telling you that the probability that they are close enough is given by the cumulative distribution function that, in principle, one can think that is an average over ensemble of trajectories, right? So here there are some caveats related to the ergodicity of this process and how I can replace average over time by average over realizations. That is, I'm not going to enter into that for two reasons. First, because I'm not fully confident with my understanding of the problem. And second, because it's a really technical problem related to the importance of trajectories in these processes. And it's, if you are interested in that, you can either talk to Benjamin, To me, read the paper. It's a very interesting problem, but it's not that clear. Anyway, what I'm going to assume is that this works, and that is true if gamma is small. So if my interactions are weak or my population is diluted, this is correct, okay? If not, not so much. Anyway, when I compute this quantity here, and I compare it with the encounter rate that I would get for a mixed system, which is this thing here, where now this gamma is the total system size, then what I get is that the ratio between these two quantities is extremely complex. What does it mean? It means that sometimes having a spatial structure makes animals interact more with each other, and sometimes it makes less. And this is an extremely risky behavior, because it's not that I can say, when I have well mixing, I'm always underestimating encounters, so I'm just careful and I just, no. It's really complex. It depends on the context. Okay, it depends on the spatial structure. That's what it means that sometimes this ratio is above 1 and sometimes it's below 1. I think it's off. Or not working. No, it's not working. That's the one that is broken, yes.
Yeah, so it's a naive question, but are you using the conditional probability or is it the joint probability?
Is the what?
Is it the joint probability or is it the conditional probability?
It's the conditional probability, you know?
But then, but the second step shouldn't be.
Wait, is the joint probability?
It's the joint. Okay.
Yes, it's the joint probability.
Okay, because I thought you're using successful attack given.
No. It's
the joint.
Okay. Yes, I think it's a joint probability.
I think there's a typo in the first line.
Yes.
Okay.
No. Yes, it's the joint probability.
Okay.
Yeah, sorry.
Thank you.
It's not a naive question. It's a poorly written slide. That was me trying to formalize things that we didn't. It's interesting when you don't come up to this result following this path. But it's the joint probability. So basically what I was saying, that when I compare these two quantities, the fact that sometimes the spatial structure makes encounters being stronger and sometimes weaker than when I don't have a spatial structure already tells us that we need to understand this problem more carefully. No, it's not that we can just say, it's always, I don't know, higher, so I need to be careful because my population size will always be underestimated and so on and so forth. So why is that behavior there? It's very simple to understand with this cartoon. So when I have a well-mixed system, my two animals are all over the place, so it really doesn't matter. The encounter rate is always the same as long as my system size is the same. But when I have these localized probability distribution functions for the position of the animal, it will really depend on how they are located in space. So when they are very close to each other, the animals will be more likely to interact than what they are in the well-mixed system, right? Because there is a strong overlap between distributions, but as they move apart from each other, then the probability is going to be lower. So there is more, just playing around with these spatial scales, it's easy to see that the process is context dependent. So just to finish quickly, because I don't have so much time, then the question is, how do these bias appear in real populations? So how can I measure this in populations? And I think that's the question that I found more interesting, not only doing the math, but also trying to find these signatures in real data. And to do that, I'm going to show you data from tapirs here in Brazil, in El Pantanal. So this cute animal is a tapir, a Brazilian tapir. This is where the population that I'm going to show you lives, and this is the animals that we have tracked. I think it's like 41 or 42 animals. And here I'm showing you the equivalent to the wolf distribution that we were seeing before, basically, but for 42 animals. Here I have a mess of data, and it's really hard to understand whether animals are interacting more often with each other or not, and it's a little bit of a mess. But what we can do is we can compute these encounter rates that I was showing you before. Now we have the expression, we have the theory, we can do it. And when you do that, what you see is that the population has a particular, and this is extremely preliminary data, but it already tells us that the population has some structure. So there are some animals here in the bottom right corner that are more likely to interact with each other than others, and there is some sort, we can construct a network of interactions between these animals just based on these encounter metrics that I was showing you before, right? That tells us how far we are from well mixed. If all of these were the same number, then my population will be well mixed. Great, so we have this quantity that tells us how frequent animals interact with, how often animals interact with each other. It's based on data. It's based on quantities that I can measure in my trajectories. So the only thing I have to do is I have to put colors to animals, and then as much as I can fit movement models, I can measure these interaction metrics. But then the question, and with this is what I want to finish, is how these master equations that we run last week, and how, more specifically, the mean field equation that we derived when everything is well mixed and deterministic, it changes when we have a space. So to do that, we can extend the individual level model that we had before, but put it on a space. So basically, I'm going to have a population of animals. They reproduce. So basically, this animal here will divide. Here, I'm not going to take into account sexual reproduction and things like that. It's a very simple birth-death process. And whenever one animal reproduces, it's going to throw the newborn somewhere that is given by a dispersal kernel. Then animals are also going to die. When they are close to each other, they have a probability of dying that is given by this rate, where we have some baseline mortality plus some density dependence here. So the more animals you have around you, the more likely you are to die. And finally, we have movement, which is where we are going to introduce this Osten-Uhlenbeck stochastic process. So without movement, this spatial birth-death process that was already introduced in the 2000 by Richard Law and Ulf Dickmann, and they even worked some analytical approximations to it. The really interesting new ingredient here is that we are introducing movement, and we are introducing the movement that we see in data. So this is not Brownian motion. Brownian motion is also done. This is the movement that we see in data. And of course, I'm going to assume that this area that the animals move over is the same for all animals, because if not, this would be a mess.
Why, when they're close together, the chance of dying is higher?
So you can imagine that these animals eat grass. And when there are 15 animals eating the same patch of grass, they have less food per each of them. Okay? Good.
Is there some benefit to adding some, basically some benefit to clustering as well? Like
we don't consider that.
Okay.
It could be. Some species are social and they just like to be close to each other. For example, the Ali effect that we saw last week is a consequence of that. But remember that what I'm trying to do is I'm trying to extend this simple logistic model to account for real movement. And in this simple logistic model, I don't have that ingredient.
Okay.
Okay?
Thank you.
So this is a individual-based model. The first thing you do with these individual-based models is usually you throw them in a computer and try to get a sense of what's going on. And that's what I'm going to show you. So what you see now in the screen is the total population size, so how many animals I have at the end of my simulation, as a function of the home range area, as a function of the spatial scale at which this semivariance function saturates. Okay? And different curves are for different dispersal ranges. That means for how far a newborn is thrown. So you see already that this pattern is a little bit complex in the sense that there is no, some curves are even non-monotonic with the home range size. The first thing that we see is that, I think this is pretty intuitive, when the home range area is very large, comparable to the system size, everything converges to well mixing, which is good. That is good. But then we have here some complexities that I'm going to break for you in a little bit fast. So the other limit, in which the home range areas are very small, you could think of it as the limit where my animals are plants. They don't move. They are stuck in the middle of their home range. So I would expect that this area here is limited by a space, right? So in this area, if my animals have very small areas, they basically should not overlap too much with each other. And that's why if they are able to go far away from their parents, the population size grows a lot. To test that hypothesis, what you can do is you can renormalize everything by the area of your environment, and you will see that in this limit here, you have a pretty nice scaling law with home range size. Why? Because there is no overlap between home range sizes, right? It's basically you're packing your population. And it is in this intermediate area where things are more interesting, because all the scales are comparable, and it's really the interactions between home ranges that is driven the population. This is not the typical finite size effect that if I go to very large systems, these two regimes collapse. As long as you allow for spatial scales to be comparable in some regime, you have this behavior, okay? So question is, well, this is a very interesting pattern that I see in the simulations, but the question is whether we can understand this mathematically. It would be really cool if I were able to, given the movement parameters of my model, of my animals, I could tell you the number of animals, right? That would be really cool. If I had an expression for my current capacity as we had before, just with the movement parameters. Because otherwise, if not, I would need to run these simulations, and that's not so fun. So a spoiler here is that yes, you can. Sadly, I don't have time to show you how you do the whole upscaling of this model, which, to be honest, was basically the goal of the lecture, but it's fine. Don't leave the goal of the lecture for the end. That's take-home message. But I can give you some hints on how you do that. And okay. So the first thing I have to do is, from the individual level processes that I have, I need to define the demographic rates of my system. So the same way before I had this division rate B, death rate D, and so on and so forth, now I need to define birth rates that are going to be functions of space. For example, this is going to be the probability per unit time that a new organism will land at position X because one organism at position XY reproduced and throw an individual, right? So this is the per capita reproduction rate, and this is the dispersal kernel that gives me the probability that the animal lands at the new position. I can do the same for the death rate. This is the death rate of the individual I. So basically, the probability that this animal dies is given by some baseline mortality plus the contribution with the interactions with any other animal in my system. And in principle, this is it, because I'm interested only in birth and death. I'm interested in population size, and movement does not change the population size. How is movement entering into my system here? Movement is determining the distance between animals, and therefore is regulating the death rate. But if I am going to write a balance equation for the density of organisms in my population, this would simply be B minus D times N, right? Where D is a nonlinear function because it depends on the sum over all the individuals. Okay. So I'm going to share with you the notes where I open up all the calculations and how you get this equation that I'm going to write now. I don't have time to introduce everything you have to know to do this. But basically, the idea is that here I do have a spatial structure, right? Here I have interactions between animals, and this is going to be given by the correlation function of my population. This is the key ingredient here, the correlation function of the spatial distribution of animals. And what is even more interesting is that this correlation function that is given by the position of the animals, in principle, since I know how they move, I can reduce it to the per correlation function of the location of their home range centers, which is quantities that I have for data. So graphically, what you can think is that if this is my simulation landscape, and I have here all my animals moving around, at a given time, if I take a picture, I will have a point pattern of my animals, right? I will have a spatial distribution of points. These animals, they will have some mean position that we introduced before as mu. And they will also have some home range size that in this case is going to be a parameter of the model, but in principle, you could also obtain it from data, right? So in the notes that I'm going to share with you, once you open up all the calculations, you will see that the equation that you get for the number of particles, for the density of animals is this thing here, where I'm going to explain you what is this integral, where the integral has two components. One is the correlation function for the position of my animals. This is a function that goes between 0 and minus 1 and plus 1. So when my population is well mixed, this is equal to 1. And this is the competition kernel that I introduced here. So this term here is what is the real correction factor that I am getting to what otherwise would be my logistic growth model that you are all familiar with. If my population is well mixed, that means that this function here is 1 for every distance, every displacement, and therefore this integral is equal to 1. This competition kernel is normalized, and then I recover the logistic model. If you can compute the fixed points of this equation, if this correlation function, if this correlation function is greater than 1, meaning that my population has a structure has an aggregated structure, this denominator is going to be larger, and my population size is going to be smaller than compared to well mixed. So this correction factor here is really tuning the strength of the interaction coefficient to tell me if the population is larger or smaller than what I would expect in a well mixed scenario. That is the key message here. The key message here is that when I have some spatial structure, the correction factor that I get for my logistic model that explains me the temporal pattern of population growth comes from the correlation function between the positions of the organisms. This correlation function for the position of the organisms is not very practical in the sense that I would need to be tracking my animals every single time. But you can transform this equation so this correlation function, instead of being for the position of the animals, it's for the mu's, for the mean position of the animals. And when you do that, you are able to write this equation in terms of parameters for which you have perfect access from data. Because when you track your animals, these two things, you can estimate them very accurately. We already saw how to do that. So basically, the spatial structure, if I track my animals for 1 year, let's say, I can get estimates for these parameters, and I can obtain this, and I can try to get an understanding of how my population is regulated by space. That is the key message here. Between this and this, there are 2 paths. One is the easy, not very, it's rigorous, but it's an easy path that gives you the information, but not all of it, which is the one that is in the notes, that I didn't open up because the time I had left was very short, and it implies introducing a lot of techniques to treat a spatial point pattern analysis. If you're interested in these kind of patterns, we can talk about them. Are completely different to Turing patterns, in which, or to the patterns you've seen last week, where you have a density field and PDEs. These are points in space, and you have spatial statistics of these points. Also very relevant, in my opinion, more relevant in ecology than continuous patterns, because usually things are discrete, are there. And writing equations for the moments of this spatial pattern, understood as a distribution of points, you basically get this equation here. As I told you, I'm going to share you the notes. I was very careful yesterday to open up all the steps and explain everything in my notes. That way, whoever is interested in this looks at them and we talk. Whoever is not gets the idea that when you have a spatial structure in a logistic model, and this applies to every system that has a law of mass action modeling the interactions, which are nearly any model in population dynamics, in epidemiology, and in many, many systems. If you have a space, the way a space enters here is in these correction terms, in terms of correlation functions. So I'm showing you this expression. I'm telling you that it's very helpful, but just to fully convince you, it would be really cool if I could set up a simulation. Imagine I have my IBM. I'm going to throw it in the computer. I know the parameters, and I can replace them here. I don't do the simulation. I just replace the mean value of each position of my animal. I know the size, and I throw those quantities here, and I get a prediction, let's say, for the total number of animals that I would have. And then I run my simulation, and I get the number from my simulations. That's the way we try to validate this approximation, and as you see, it works pretty well. Just to emphasize, here I'm not computing anything in terms of the position of the animal. I'm computing it in terms of the statistical pattern of movement of the animal. That's important, right? It's not that I am tracking the position at every time and then I'm using the same information twice. I'm just using the statistical properties of the movement. Okay, so that's all, because I don't want you to kill me for a piece of cake. At least if you're going to kill me, let's make it for something important. Just quickly, on Thursday, I will give one of these Papos de Física in Cervejería Nacional here in São Paulo. I don't know why they make me talk in a bar. I may think maybe something is going on there. It's in Portuguese, unfortunately, for those of you that don't speak Portuguese. Maybe you have to drink more beer than those that speak Portuguese. It's not part of the school. It's just in case you want to come. I'm going to talk about patterns in the way that Johan was talking. And I'm going to show examples from Brazil. I'm going to tell a story on how termites engineered a landscape of the size of the UK in Bahia, here in Brazil, and what is that these termites are doing in that ecosystem. And I have a PhD position open in the group in Görlitz, so if you are interested in pattern formation, either this way or reaction diffusion way, we can talk, and I would be very happy if you apply. We are based in Görlitz, which is a very nice town in East Germany that looks like this. If you want to know how Görlitz looks like, you can see Bastards Inglorious, Inglorious Bastards, or Hotel Budapest, or any of these World War II movies, because all of them are recorded there. It's called Görlitz, so it's quite funny. So this is how the village looks like. This is also how the beach looks like. We have a beach. For those of you that are from northeastern Brazil, please don't compare, because this clearly outbeats any beach in the world. And we also are close to hiking spots, and yeah, we do try to do cool work. So if you are interested, just let's talk to me, and and we can discuss. And if you have more questions, of course, please ask. And thank you.
Thanks, Ricardo. That's very nice. So you have a very non-well-mixed situation, but you can express the dynamics in terms of a well-mixed kind of equation with a rescaled competition gamma parameter, right?
Mm-hmm.
Why does this work like this?
Why does it work? So it works because, well, so I honestly think it works because it's a correct description of the system. So that is the most important thing. I think it's actually how the system, I mean, it provides the proper description of the system. It's not that it's not the same, no, that your theory reproduces the system, then it describes it properly. If you want to have intuition on why this description is accurate, it's because when you, it's because when we write the equation for the dynamics of the number of animals, we do keep track of the spatial correlations all over the derivation. So when you write the equation of, you can think of your, you can think that you have this distribution of animals, and you can define a point pattern that is the sum over I of a lot of deltas. This is the way you define usually these point patterns, right? In that case, this density of organisms is just the first moment of this thing. When you want to calculate the dynamics of this moment, it will depend on these two things here. And here is where you have spatial correlations. If you keep track of these spatial correlations throughout your derivation, at some point, you will get how they enter into the equation, right? So usually, I don't know how many of you are familiar with the study of these patterns and spatial moment dynamics and things like that. But usually what you would do is that you would write an equation for the number of particles, and you would also write an equation for the second moment of the distribution. That basically gives you the pair of densities, the second moment, the covariance between the number of pairs at a distance displacement c. So usually what people do is they solve these two integrals together, two equations together, to get an idea of how they co-evolve, let's say. The problem is that when you do that, and that's why I said that there are two ways of doing this calculation. One that is the one that I would really like to do, which is deriving the full theory for a bunch of particles in harmonic oscillators with thermal noise, which is what this model is telling us. When you want to do this, for this system, it's really hard, because our particles, they are not, they are distinguishable. Each of them has a different focus, and then this quantity here is not spatially invariant, and that makes the calculation really, really hard. So instead what we do is we do this, we write the first equation, we keep track of the correlations, and then we try to write the correlations in terms of the parameters that we know. That's more or less the steps of the calculation. But why it works? Because it keeps the spatial structure and it does it with accuracy. I mean, I don't know if it doesn't work all the time, huh?
Yeah, if you start from a Fisher equation, for instance, like you have spatial terms like diffusion or something like that, and you integrate this equation, there is a competition term there that you cannot actually integrate and get the. So something will still depend on position, right?
Yeah, but the Fisher Kolmogorov equation has, as any other reaction diffusion equation, the only thing that it does is that you remember the definition that Nagel gave for this gamma omega here? He said, this is not the system size. He said, this is the correlation volume. When you write an ODE, this correlation volume is effectively the system size. You want to think about that in this way. When you write a reaction diffusion equation, all you are doing is assuming this thing in a smaller size. That's why, even though some people don't like it, I like to think about deriving these PDEs in a lattice, because that's what you are basically doing to build intuition. You're assuming that your interactions happen within a lattice site. And then you take that limit to 0 and you define a density. But within the volume, you're assuming a high density anyway, right? We are not doing that. This is discrete. The simulations are with 200 animals, 300 animals. It's low numbers. That's why writing fields here is not that easy.
Yeah, thank you.
Thanks, Ricardo, for your lecture.
Great
Lecture. I have a curiosity about, you are thinking about to modeling this, to use this model to modeling with kind of system. I think because I wonder if you are think about to modeling movement of birds, for example. We have collect motion between birds. How we can adapt this model to take into account this behavior, like birds, like, for example, fish. I think this is missing is this model.
Yeah, so there is something that is very important, that is time scales. So when you think about flocks, for example, the time scales that people studying flock are interested in, usually it's a good assumption, is reasonable to assume that the population size is fixed, right? You are interested in time scales that are not comparable to the lifespan of the birds. So I don't think, I mean, introducing those things here, I mean, could be an interesting exercise. Where I think it would be more relevant is in what I was showing here. Here. Because when we do that, that's something I didn't say. When we compute these encounter rates, we assume that the animals are independent from each other. And that is a big assumption. You can always write your theory, and that's something that is not that hard. Assuming that these two stochastic processes are cross-correlated. So there are covariates between the position of the animal 1 and 2. You can compute this encounter rate, and you can calculate how the encounter rate changes with the cross-correlation. That's not really that hard, provided that you are reasonable in your correlation structure. And then you see that when they are positively correlated, the encounter rate goes up and down. Or even in the case of flocking, for example, where you have alignment, if you have this alignment acting when particles are farther apart from the encounter rate, sometimes they don't even encounter, no? If they are flocking that way. So my short answer would be that I think for population dynamics, so flocks operate at a much smaller time scale than the questions that we are interested in, but you can introduce them in encounter rates. What is really hard is once you have the theory with correlations to do the statistical estimator. That we haven't been able to do that yet. We want to, we are trying to, but we haven't been able to do it yet. It's because the problem gets 4D instead of 2D.
Shall we continue the discussion over coffee break?
Yeah, thank you.